Hello, my name is Poonam Mehra and I'm an EMBO long-term fellow at University of Nottingham. I basically work on studying root adaptive responses under transient moisture stress. And I'm very happy to give a brief overview of our recent experiments conducted at the aeroponics platform at UCL Louvain. As we know that soil is a very complex matrix where water is heterogeneously distributed, and when roots pass through this variable environment, they show adaptive response. Like when roots pass through an air gap, they don't form lateral roots, and we call this as zero branching. So to dissect the molecular mechanism behind the zero branching response, we plan to use a diverse set of criteria. So we have a panel of 309 criteria accessions which are sequenced, and we have a very high quality sequencing data available for this diversity panel. So we decided to impose a transient moisture stress on this criteria panel to find out the molecular mechanism behind this response. To conduct this experiment, we are highly thankful to EPPN 2020 and the UCL Louvain team, which helped us to impose transient moisture stress on Citeria. So we use this platform to perform very high throughput screening of different root traits, especially lateral root density and Citeria under transient moisture stress. Very uniform germination of seedlings was easily achieved on this aeroponics platform. The identity of each genotype was clearly maintained, and since the platform is so big that proper replication could be easily achieved. Transient moisture stress was imposed both on the primary roots as well as on the nodal roots. Water was withdrawn for a few hours followed by normal watering, and root images were periodically captured by the camera to study the effect of stress on different root traits. Wealth of phenotypic data set was generated by automated imaging platform. We now plan to use this dataset for downstream GWAS analysis to identify candidate genes involved in the zero branching response. We also plan to identify the genetic loci for different root traits in Citeria. We would like to thank the UCL Louvain team, especially Xavier Dre, Olivier, and Thomas, who made this experiment possible during the challenging times imposed by COVID pandemic. We would also like to thank our collaborators in the project for actively engaging with the UCL team through various online meetings which guaranteed the success of this project. And finally, I would like to thank the EPPN 2020 for funding us this research and providing us the access to the state-of-art aeroponics platform at UCL. Thank you.